Welcome everyone to my talk, Multimodal Membrane Adsorbers for Clearance of Minute Virus of Mice. My name is Patra. Before proceeding further, during this talk, I would like to introduce the co-authors. I'm working as a postdoctoral fellow in the Department of Biomedical Engineering with PI of this project, Professor Xianghong Qian. And Shouting Chen is a PhD student in Department of Chemical Engineering working with Professor Ranil Wickramasinghe. So the multimodal membrane adsorbers are being investigated during the last decade for their potential application in the downstream processing industry. In the capture step of the standard antibody purification platform, protein A chromatography is in generally used in the bind and dilute mode to provide a final product purity of more than 95%. However, in the polishing step, ion exchange chromatography and hydrophobic interaction chromatography are employed in the flow-through mode to obtain host cell protein purity of less than 100 ppm, DNA removal of less than 10 ppb. However, all these steps have a major disadvantage of the significant amount of buffer makeup volumes and limited chromatographic capacity, which contributes to significant amount of cost in the downstream processing part. During the last few years, a lot of effort in this aspect have been made to develop the multimodal membrane adsorbers, combining the different types of ligands which could employ the different interactions of the ion exchange or the hydrophobic interaction in a particular membrane. However, application of those multimodal membrane adsorbers will need significant amount of process optimization. So in the scope of this study, we have been working with Purilogics LLC with their newly developed multimodal membrane adsorber Purexa for their application in the virus clearance. So in this study, we have rigorously investigated the effect of buffer pH or flow conditions to optimize the virus clearance performance of the multimodal membrane adsorbers. Also, we have rigorously investigated the effect of model proteins like PSA, cell proteins sliced from CHO cells, which we'll call CHOPs in this talk. And also, we have studied the effect of IgGs on the overall performance of this multimodal membrane adsorber. Based on all these results, we are trying to develop a phenomenological model which could describe the MBM binding using this multimodal membrane adsorbers. So in this study, we have used mainly 3 different membranes. One, the Purexa, which has been the center of this study. And we have tried to compare this performance of Purexa membrane with 2 other commercially available multimodal membrane adsorbers from 2 other companies, which we will call membrane A and membrane B. So here a comparative membrane characteristics show that the multimodal membrane adsorber which we have used, Purexa, has a membrane volume of 30 microliter as compared to the membrane volume of membrane A and membrane B, which are 80 microliter and 200 microliter. The other properties are comparable here. However, the BSA binding capacity as investigated from a particular study with the BSA solution with 3 gram per liter and trace buffer conditions at pH 7.5 were higher than, at least 2 times higher than the membrane A here, 114 milligram per milliliter. And although the performance was lower than membrane B, which is 200 milligram per milliliter. Interestingly, the membrane A has a ligand type of primary amine, and membrane B has a quaternary amine ligand as documented. So here in the particular membrane interaction study with the model proteins like BSA was performed using BSA solution of 3 gram per liter as described in the earlier slide, with 20 millimolar tris buffer at pH 8 and 125 millimolar sodium chloride as a loading buffer. The dilution buffer contained 625 millimolar sodium chloride. Under this set of conditions, the fast protein liquid chromatographic study was performed for the loading of 3 gram per liter of BSA solution on the pyrrologist membrane. And the studies revealed that under this set of condition, the other two membrane had a dynamic binding capacity of 10%, which is in generally a parameter used to characterize the membrane performance with respect to the model proteins. So the values are comparable with the other commercially available membranes. Similar studies were performed for the characterization of this particular membrane for the whole cell proteins obtained from CHO cells, the CHOPs. Here in a CHOPs concentration of 2.3 gram per liter was used under similar flow conditions, the buffer conditions. And the 10% dynamic binding capacity for the CHOPs was found to be 25 milligram per milliliter, which was further used as a to characterize this multimodal membrane adsorbers. As described as one of the main research objective of this study was to tune the multimodal membrane adsorber performance under different buffer conditions and the pH conditions. So we have rigorously investigated different risk buffer conditions with different pH varying from pH 9 to pH 8, 7.5, pH 6. Herein at pH 6, the membrane performance in terms of virus removal, the log reduction value was obtained to be the lowest, that is 3.14, greater than equal to 3.14, which could be attributed to the isoelectric point value of the embedded particle, which is 6.2. That significantly shows that the multimodal membrane adsorber has electrostatic interaction, which contributes to the lowering of the log reduction value below 4 logs per milliliter, which is an acceptable value as obtained in the range of pH 7.5 to pH 9. So for pH 6 conditions, this particular multimodal membrane adsorber under the tris buffer shows a decrease in the log reduction value, and that is the decrease in virus removal performance. Similar experiments were performed using the phosphate buffer conditions in the pH range of pH 7.4 to pH 8. However, in this pH range, no significant effect in virus removal performance was observed, and more than 4 logs per ml virus removal performance shows a steady performance for this multimodal membrane adsorber in this pH range under phosphate buffer conditions. Further, to investigate the effect of other protein molecules, a standard protein concentration of BSA 50 ppm was used under 2 different pH conditions to investigate the effect of this BSA in the overall virus removal performance for this particular multimodal membrane adsorber. Similarly, at pH 6, the competitive binding was observed for a BSA concentration of 50 ppm. However, as the BSA concentration was increased to 3 gram per liter, almost no virus removal was observed. That means the limit for this multimodal membrane adsorber for BSA concentration will be 50 ppm. And the pH range will also have to be in the range of greater than higher than 6, pH 6. Similar investigations are performed for the CHOPS. The CHOPS concentration of 50 ppm, so the steady membrane performance for the virus removal under both the buffer conditions, stress buffer conditions at pH 7.5 and phosphate buffer conditions at pH 7.4. However, as the CHOPS concentration was equal to 500 ppm, the membrane performance for the virus removal obviously decreased significantly. Further, IgGs were used to investigate their effect on the virus removal performance, as evident from this data that IgG concentration of 1 gram per liter to 5 gram per liter under different pH conditions showed significant effect on the virus removal performance. Herein, the PI value of the IgG samples were used were in the range of 7.2 to 7.5. So based on this data, although the membrane performed significantly well for the other impurities, the IgG showed significant effect on the virus removal performance. Overall, a comparative study on the membrane virus removal performance was performed with other two commercially available membranes, membrane A and membrane B. Herein, the loading concentration was used 20 millimolar trace and 125 millimolar sodium chloride. And the elution buffer condition was used 20 millimolar trace and 625 millimolar sodium chloride. The Purexa membrane showed steady performance under this set of conditions for the virus removal. Overall, if we compare the virus removal performance, these newly developed multimodal membrane adsorbers were as efficient as the other commercially available membrane adsorbers with a log reduction value of 4 logs per ml for different buffer conditions and pH conditions other than pH 6. The particular membrane adsorber used in this study showed steady performance with a BSA concentration of less than 50 ppm. Similar trends were observed for CHOPS concentration of less than 50 ppm. Here in the IgG concentration of 1 gram per liter to 5 gram per liter significantly contributed to the lowering of the ambient removal performance. So based on all these results, we are trying to tune the ligand density and the other parameters which could help in terms of fabricating a better multimodal membrane adsorber, which could be applied in the polishing step. And overall, this membrane adsorber could be used for most set of conditions, but still needs some improvement as obtained from this study. For this work, I would like to acknowledge Jinxiang Zhao and Daniel Hen from Purilogics LLC, who have been constantly in touch with us in terms of the different investigations. I'd like to thank the University of Arkansas for the financial support and my lab mates, Namila, Da, Wenbo, and Hong for their help during different experimentation. And also we'd like to thank the fund from AIChE SBIR for this project. I'd like to thank everyone for listening to my talk. If there are any questions, I'll be available in the question and answer session. Thank you.